Welcome. In the last video, I told about pollen-pistil interaction. I'll be continuing even in this video about that pollen-pistil interaction. And after this pollen-pistil interaction, I will be discussing about double fertilization, how it takes place in case of angiospermic plants. So now let's begin this video with the pollen-pistil interaction. So in the last video, we understood that the pollen grains will come and settle on the stigma, and of course we also understood that not always the right type of pollen will come and settle on the stigma. So there will be various types of pollen grains from the same species, from the same flower, right, and from different genera also the pollen grains will come and settle on the stigma, right. And in this case, right, so this stigma will be having the ability to recognize whether that is of right type or not. So this pollen pistil interaction is actually a dynamic process. So it's a dynamic process, right. And once the pollen is recognized, so it will allow whether to germinate, or it may also stop the process of germination, or stop the growth of pollen tube in the stigma and right style. So after the pollen recognition, so I say, so once the pollen is recognized, so there will be two things that can happen. So there is a promotion, or there can even be inhibition, right. So if it is the wrong type of pollen, then it will be inhibited. So this inhibition will be seen in case of the wrong type of pollen. So I told that mostly the plants will go for cross pollination, and they like to accept the pollen right, coming from the, what I say, different plant but belonging to right same species. So this is a right type. So it's a right type of pollen. So which means that this is a pollen from another flower. So it's from another flower, okay, from another plant but of same species. So but of same species. So the pollen that comes from another flower that belongs to another plant, right, but belongs to the same species, will be of the right type, and whatever that pollen, other type of pollen, will be considered as wrong type. So in that case, those pollen grains will be rejected. In fact, their growth will be inhibited. So if already they germinated, right, the pollen tube growth will be stopped. So that's what I said. After the pollen is recognized, so there are two things that can happen. So one, it may promote the growth of the pollen grain, or it may stop that growth. Okay, so in this case of promotion, then it leads to what I say, the pollen germination. It leads to pollen germination and pollen tube growth, right. That pollen tube will carry the male gametes finally to the embryo sac. Okay, so where of course the further fertilization, which we will see later how it takes place and all, right. So the pollen is after recognized, so it has two things: either it is promoted or inhibited. And it is inhibited, okay, if it is of a wrong type of pollen, and it is promoted if it is of a right type. And I told that the right type is the case where the pollen from another flower of another plant but belonging to same species, right. Now, understanding this pollen-pistil interaction by a plant breeder is a very important. So the plant breeders who are involved in the production of hybrid plants. So here the plant breeders need to understand this pollen-pistil interaction. So how what I call the stigma is playing a role in identifying the pollen and accepting the pollen, or whether it is rejecting the pollen. So all these things are very important you know for production of the hybrid plant. So this will help in the production of desired hybrid. So understanding pollen pistil interaction, pollen pistil interaction is important for developing desired hybrids, right? So if they are able to understand this mechanism properly, then the plant breeder can even make a pollen that comes from a different species or from a different genera also to get germinated. So right, so here say this is the stigma, and here the plant breeder have taken the pollen from different species. So he has taken from different species, or taken from different genera or different genus only. So from different genus he has taken, or from different species he has taken that pollen, and he can make that, what I say, this pollen to get germinated on this stigma, right? So that will help us to produce something called the desired hybrids. Means these are the hybrids which have qualities that will help us to give more yield. So of course the ultimate point in obtaining the desired hybrids is that to get more yield. So a farmer wants plants that can give more yield. Okay, so here for that, let's understand by taking an example. In this case, say there are two plants we have. So we have is plant A and plant B. Now in case of plant A, so this is the one that produces of course good yield, but the point is that it is not disease resistant. So it is not disease resistant. So if we can make it disease resistance, then we will be able to get a very much good yield, right? And we have another plant. In this case it produces less yield, so it's not that much good at producing, but this one is resistant to disease. So I can say this is disease resistant, right? So now if I combine these two characters, so that is ability to produce good yield and make it a disease resistance, then I will be able to get a crop with the desired character. So here in this case we get a crop with the desired characters. So what are the desired characters? So these two are the desired characters. So ability to give a good yield and be disease resistant. So in that case it will be able to give you a very much a good yield. So I say that plant which has these two desired characters as a desired hybrid. So that one is a desired hybrid. Okay, so these plant breeders need to understand this pollen pistil interaction, so that it will help them to produce these desired hybrids, right? So finally I can say that this crop which is the desired hybrid is a a commercially superior variety. It's a commercially superior variety, so that has a desirable characters. Okay, so there are certain instances where we can see many plants who have some or other problems, so we like to develop the crops, okay, that can give us the maximum yield, right? And all the plants will not have all the desirable features, so we need to cross different plants to get desirable characters in one plant, and that becomes something called the superior variety. Okay, so this is how a plant breeder develops these superior varieties. Okay, fine. Now the point is that how they are going to make this, of course, understanding of this pollen-pistil interaction will make them to produce the desirable hybrids, but how they perform it. So here, let's understand that. So to produce these desirable hybrids, right, so we need to take the pollen from the desired plant and put it on another plant which we have chosen as parents. Say, so we have chosen two plants as parents, so this is plant A and this is plant B. So these are parents with desirable characters. So plants with the desirable characters. So now I want to combine the characters present in the plant A with the characters present in the plant B, and then finally produce a superior variety. Right, so let's take the same example which I have taken earlier and say that so it can produce good yield, but it is not disease resistant. Okay, so this one, right, doesn't produce good yield. Okay, and but this is very much disease resistant. So this is disease resistant, right? So I want to club these two characters and make it a new superior variety. So that's what we call a desirable hybrid. Now what I say here, say in both the cases, both the plants, they are producing bisexual flowers. So here they are producing bisexual flowers. Okay, so in this case also there are bisexual flowers. Okay, okay, so this is in this case the bisexual flower, and this is another bisexual flower. Okay, so now point is that if the both plants are bisexual, right, so I should use two methods. One, it is called emasculation, and another process is called bagging. So these are the two steps, right, which will come under something called artificial hybridization. So to get the desirable hybrid, I need to perform artificial hybridization. So what we mean by this artificial hybridization is the transfer of pollen grains from the anther to the stigma of another plant, okay, manually. So we transfer that pollen from a desired plant. So from this plant A, I am going to collect the pollen grain, okay, and I will be dusting it on the stigma of right, another plant, so which I have chosen, right. So here it is not like randomly taking place, right, either by wind, water, or some other animals bringing the pollen, and in that case we cannot come to know what are the parents of those offsprings. But in this case, whatever the offspring which I am going to obtain, I clearly know that what the parents are, right? So here the pollen grains of one plant, right, they will be transferred, okay, manually from from this plant to the stigma of another plant. Now to perform this, okay, we have to do two things. One, it is emasculation, and another one is bagging, right? So now let's see what actually is this emasculation process. So here the emasculation process is the removal of anthers, okay, from the flower bud before it, okay, dices by using forceps. So we need to remove that anther. So in this case, say in case of plant B, I need to remove the anther. So the point is that the pollen grains from this anther may fall on the, what I say, the stigma, and it can result in the process of fertilization if it is what I say, a self-pollinating plant, right? So whatever that the situation, so in this case I need to remove these anthers from the flower. Means the flower, even before it opens, I need to remove these, what I call, the anthers, right? So that process we call it emasculation. So removal of anthers, okay, from the flower bud before it dices by using forceps, right? So now let's see in this case, okay, so say this is the flower that is not opened yet, okay. So this is a flower bud, right? Now what I'm going to do, right, the first step in this case, that is artificial hybridization, is emasculation. So what I'm going to do is remove these anthers. So slightly I will, okay, open this flower and then cut these anthers and completely remove these anthers. So now the process of emasculation is completed. So once that emasculation, okay, completed, now I need to cover this flower bud with a, right, a bag, usually that is made up of butter. So I can say like a butter paper. So this is a butter paper. So this is a butter paper. With that butter paper, I'm going to completely, right, close that flower. Okay, so this process, what we call it bagging. So I can say in simple words, bagging is covering of okay, emasculated flower with a bag of suitable size. So covering of emasculated flower with the bag of suitable size is what I say the bagging. Okay, so in this case this bag is made up of okay, butter paper, and this will actually help in contamination of this right pistil with the unwanted pollen. So I don't want any other pollen from other sources to come and settle, because I already decided that I need to transfer the pollen of this plant A to this, okay, stigma. So for that process I have covered this flower once the process of emasculation is completed. Of course this emasculation is not necessary if the female plant produces unisexual, okay, flowers. So if female plant produces unisexual flowers, if female plant produces unisexual flowers, okay, so then there is no need of emasculation. So because already on the, say in case of female plant, it will have pistillate flowers. So there is when there are pistillate flowers, and we know that a pistillate flower will not have anthers. In fact, stamens are not present. So in that case, right, there is no need of this process, what I call emasculation, right? So if the female plant produces unisexual flowers, then there is no need of emasculation. But the point is that bagging is required. Okay, so before the flower opens, so you need to cover that flower with the help of a bag which is made up of like this butter, so butter paper, right? So I'm sorry, one small correction here. So I don't mean that the butter paper is made up of butter. So here what I mean is that the butter paper is used to cover the flower bud after the process of emasculation, right? Now once this flower, so from the plant A, it starts producing the pollen grains. I'm going to collect these pollen grains, okay. So from this plant A, so these are the pollen grains, okay. So these pollen grains from the plant A, so I'm going to transfer it onto the pistil of this plant B. So for that process, as I told, this flower bud after the emasculation process, it is covered by the butter paper. So I'm going to remove this, butter paper. So now the point is that, okay, of course by this time this flower is mature. Okay, so of course after the maturation only I'm going to remove this, okay. Okay, so now, right, so this flower is mature. Now once this is seen, now this pollen grains, I have dusted it on this, the stigma, and then later once this is finished, again I'm going to rebag this with the same thing, okay, so the butter paper, so to avoid unwanted pollen here, okay. So here you can see whatever that pollen from the plant A, so which I have put it on the stigma of the plant B. So this process is what we call it the artificial hybridization. So once this pollination is over, then this flower, right, later is going to develop the fruit and seeds. Those seeds are important. So we sow those seeds and see whether those seeds develop into plants that will have the characters which we wanted. So if it is the case, then we have developed, so if we see that desirable characters in the offspring, then we have developed okay, the superior varieties which have the characters of both the parents, right? So this is how we perform the artificial hybridization and are able to produce commercially important superior varieties of plants, right? Okay, so of course here I'd like to change this. So this is a flower at maturity. Now it is not a flower bud, okay. So when it was in the flower bud condition, we performed emasculation. Once the flower matured, so we are going to remove this, okay, cover, and then transfer the pollen from the plant A, which we have selected, okay, as one of the parent. That pollen will be dusted on the stigma. So once that is finished, right, we will be covering it back. So this process we call it the rebagging, okay. So emasculation, bagging, and then after the transfer of the pollen, then you have to rebag that flower, and that ensures, okay, the pollen from the plant A will only be fertilizing those ovules inside that flower, right? So okay, so that's all about this pollen pistil interaction. So now let's move on to the fertilization part. So whatever this part we have discussed is the pre-fertilization events. Okay, so now move on to the next part, that's the fertilization process. So how fertilization takes place in case of angiospermic plants. So here let's see the next one, fertilization. So here in case of angiosperms, the double fertilization is unique feature okay, of angiosperms. Only in case of angiosperms we find this double fertilization. So this double fertilization, it involves actually two fertilizations. So one, it's called the triple fusion, and another one called syngamy, right? So as the syngamy and triple fusion take place, right, we call that as double fertilization. So now let's see how that double fertilization, okay, takes place. Now the right type of the pollen settles on the stigma. So we know that right, so the pistils will have this ability to find out, okay, the right or the wrong type. Okay, so once they settle on the stigma, right, so say this is the right type of pollen, it's the right type of pollen. Okay, so once the pollen is recognized, okay, so now the vegetative cell, so I think we remember that the cells inside this pollen grain, either it will be, okay, two cells or it may be three cells. So in our earlier classes we learned that the pollen grains can be released at a two-celled stage or will be released at three-celled stage. So if it is released at a two-celled stage, right, then it will have one vegetative and one generative cell. Okay, so as just mentioned, the two-celled stage, in that case it will have one vegetative cell and another one is generative cell. And if it is a three-celled stage, if the pollen grain is at three-cell stage, then it will have, okay, one vegetative cell. So in this case it is only one, and generative cell is also one. So in this case the vegetative cell is one. The second one, the generative cell itself divides, okay, by mitosis and produces two male gametes. So in this case it will have male gametes which are, okay, two in number. So that is the reason why I have mentioned the three-cell stage. So I can say in this case the generative cell, it divides by mitosis and it produces two male gametes. So it is going to produce two male gametes. So that's what we see in case of the three-cell stage. Okay, so once the right pollen, so we the stigma comes to know it, so then what happens is, right, through one of the germ pore, the vegetative cell, right, starts coming out, and that forms what is called the pollen tube. So this pollen tube will be carrying the male gamete. So you will see inside that, so these are the male gametes. Of course, the generative cell, if it is a two-cell stage, have undergone the mitosis and produced two male gametes that will be seen in case of this pollen tube. So the pollen tube develops from vegetative cell. So the vegetative cell develops the pollen tube, right? And already we know that the male gametes will develop from the generative cell, right? Of course, here you may have in front of this the presence of okay, tube nucleus. So this is a tube nucleus, means that is the nucleus of the vegetative cell, right? And we'll have this, okay, male gametes. Now the pollen tube grows through the, you know, the style. Okay, so this is the stigma, and now it enters into the ovary. Okay, so here say this is the ovule. So it passes, okay, near to the micropyle of the ovule. Through the micropyle it enters, okay, into the ovule, and then finally it enters into the embryo sac. So now let's see how that process takes place. So I hope this part is clear. Once the pollen tube germinates, right, the pollen tube carries the male gametes, okay, through the stigma, through the style, and into the ovary, and finally enters into the ovule through the micropylar end of the ovule. So now let's see at ovule level. Okay, so here is the embryo sac, and so this is the pollen tube carrying the male gametes, and that has entered the micropyle. And now we know that embryo sac will be having, so this is the embryo sac, and this embryo sac, towards the micropylar end there are synergid cells, and these synergid cells will have filiform apparatus. So they have filiform apparatus. Okay, so here is the vacuole, the nucleus, and here is the right egg cell, and here there are two polar nuclei, and here are three antipodal cells. I hope you remember all this antipodal. So this is the chalaza end of the ovule, and this is the micropyle end of the ovule, and these two are the polar nuclei. So those two are polar nuclei, and this one is the egg cell, and these two are synergid cells, right? So we know that this one egg cell and two synergid cells which form the egg apparatus, right? Now this is the pollen tube, and you can see the pollen tube, and so you can see the male gametes here. Okay, right? So this pollen tube enters, okay, through one of these synergid cell. So here one of the synergid cells starts degenerating, and the pollen tube enters, okay, into that degenerating synergid cell. So this filiform apparatus, okay, as I told, helps in guiding the pollen tube into the synergid cells. Okay, so it helps in guiding. So there is in fact a chemotaxis, right, taking place, and as a result the pollen tube, okay, enters into one of the degenerating synergid cells. So So this is a degenerating synergid cell, and I'm just showing here. Okay, so finally the pollen tube entered, okay, into it. Once the pollen tube enters into it, the pollen tube, okay, breaks open and then releases these two male gametes into the cytoplasm of the, okay, that degenerating synergid cell, right? So these two male gametes now entered, okay, into, okay, finally into the embryo sac. Now out of these two male gametes, okay, one of the male gamete is going to fuse with the egg cell. So one male gamete plus one egg cell. So one male gamete and one egg cell, okay, their fusion is what we call it syngamy, and it results in formation of zygote. So it results in the formation of zygote. Okay, the next one more we have is the other male gamete. Now in this case, okay, so another male gamete plus it's going to fuse with something called the secondary nucleus. So what happens is that two polar nuclei fuse. The two polar nuclei fuse to form a secondary nucleus. So this secondary nucleus will be diploid in condition. So I hope you remember that every cell in the embryo sac is haploid. Okay, so these antipodals, the polar nucleides, whatever, all those are haploid in condition. So now this overall is the largest cell, okay, which we call it the central cell. Okay, like now the one male gamete is going to fuse with the secondary nucleus. As I told, secondary nucleus is haploid, male gamete is, second nucleus is diploid and male gamete is haploid. Whereas in this case, okay, egg cell is haploid and male gamete is also haploid. So as a result, you will get a zygote which is diploid. Okay, right. Now the fusion of one male gamete, which is haploid, will fuse with the secondary nucleus, which is diploid. So in this case, right, it is called the triple fusion. So there is a, okay, fusion of three nuclei. So hence it's called triple fusion, and this triple fusion results in formation of okay, endosperm. Results in the formation of endosperm. So this endosperm is triploid. So hence I say in angiosperms the endosperm is triploid. So of course the zygote is diploid because it is formed by the union of one male gamete, that is haploid, with the egg cell which is also haploid. Okay, but this triple fusion is a result of one secondary nucleus which is diploid and one male gamete, that is haploid, finally resulting in the formation of a triploid endosperm. So endosperm, okay, is food for the developing, okay, zygote. So I just mentioned that here. So endosperm is food for developing zygote. So zygote is going to use this endosperm and then start dividing, and then finally it results in the formation of embryo, right? So this is the whole process of double fertilization. So as I told, the double fertilization is a unique feature of angiosperms, and as there are, okay, two fertilizations, so one, it is syngamy. So this is the one, okay, and this is the second one. So there is a, you know, process of syngamy, and there is another triple fusion. So these two together it's called double fertilization. Right, so after the fertilization, so what is seen in the embryo sac? So once the fertilization is completed, okay, we need to keep in mind that antipodals will degenerate, then synergid cells will also degenerate. And what you see here, okay, this is the embryo sac, and here you will find the nucleus, okay, and here is the, okay, what I say, the zygote. Okay, so this is the zygote form, and now this central cell, so this the, okay, as I said, is the central cell. So this central cell after fertilization is going to develop into primary endosperm cell. So now this central cell is no more called as the central cell. We need to call it a primary endosperm cell. So in short, I write PEC. And the nucleus of this is called the primary endosperm nucleus. So it's PEN. So this is triploid. Okay, of course, this one, the primary endosperm cell, will also be the triploid. So this zygote is diploid. Okay, so this is what you see after the fertilization, right? Only the primary endosperm cell, okay, with its nucleus, which we call it primary endosperm nucleus in short, or it's abbreviated as a PEN, and it will have, okay, the zygote present, which is diploid. So this is all about the fertilization. So we have completed this fertilization process. In the next class, I shall discuss about post fertilization events. Thank you.